What if the most dangerous thing happening inside your body right now is total silence? Not pain, not a warning sign, just silence, while a clock that nobody told you about keeps ticking. Here is a fact that still stops me in my tracks, even after 2 decades of practicing medicine. Scientists can now look at a sample of your blood and determine your biological age. Not the number on your birth certificate, but the actual age your cells believe they are. And in study after study, those two numbers are not the same. For millions of people over 55, their cells are aging 10, 15, even 20 years faster than their calendar age. That means a 62-year-old woman can have the cellular profile of a 78-year-old. Or, and this is the part nobody talks about, a 70-year-old can have the biology of someone who is 55. The difference is not luck. It is not entirely genetics. It comes down to a handful of biological mechanisms that are either silently accelerating your clock or quietly winding it back. I am Dr. Linda Harrington. I have spent my career studying the intersection of aging biology, cellular health, and metabolic function. And today, I want to walk you through the specific mechanisms that drive biological aging, and more importantly, what the latest science says you can actually do about them. Stay with me through this entire video, because the most important piece of information, the one that surprised even me, is not at the beginning. I have saved it for later, and I promise it will be worth the wait. Coming up in just a moment, why the food that many doctors still call heart healthy may be silently oxidizing your arteries from the inside. Let us begin with a process called inflammaging, a term researchers coined by combining the words inflammation and aging. It describes a state of chronic low-level immune activation that has no obvious cause, produces no obvious symptoms, and quietly accelerates the deterioration of nearly every tissue in the body. Here is the mechanism. Your immune system uses inflammatory molecules called cytokines, including interleukin-6 and tumor necrosis factor alpha, to respond to infections and injuries. Under normal circumstances, inflammation rises sharply, does its job, and then resolves. In inflammaging, that resolution never fully happens. The immune system stays in a low-level state of alert, continuously releasing cytokines into the bloodstream even when there is no active threat. The result is a slow, systemic oxidative environment that damages cell membranes, accelerates telomere shortening, impairs mitochondrial function, and activates a transcription factor called NF-κB, which then promotes the expression of even more inflammatory genes. It becomes a self-reinforcing cycle. A landmark paper published in Nature Medicine identified elevated inflammatory markers, particularly C-reactive protein and interleukin-6, as among the strongest predictors of accelerated biological aging in adults over 60. More telling, these markers were elevated in individuals who describe themselves as feeling completely healthy. So what drives inflammaging? 3 primary culprits emerge consistently in the research: oxidized linoleic acid from industrially processed seed oils, chronic elevation of the stress hormone cortisol, and glycemic variability from refined carbohydrate consumption. Each of these independently activates NF-κB. Together, their effect is compounding. The intervention I have found most impactful is the systematic removal of omega-6 dominant seed oils, including canola, soybean, sunflower, and corn oil, from the diet. Linoleic acid, the primary omega-6 fatty acid in these oils, has a biological half-life of approximately 680 days once incorporated into cell membranes. When it oxidizes, it produces 4-hydroxynonenal, a highly reactive aldehyde that directly damages mitochondrial proteins. Replacing these with extra virgin olive oil, rich in oleocanthal, a natural COX inhibitor with anti-inflammatory properties comparable to low-dose ibuprofen, and pasture-raised animal fats can meaningfully reduce the inflammatory burden over several months. In the next section, I want to show you what is happening at the level of your cellular power plants, and why the energy crisis inside your mitochondria may explain why you feel tired no matter how much you sleep. Your mitochondria are not simply the powerhouses of the cell, as every biology textbook states. They are also the primary regulators of cellular aging, apoptosis, and metabolic signaling. And in adults over the age of 50, mitochondrial function declines at a rate of roughly 8 to 10% per decade, unless specific interventions are made. Here is the core problem. Mitochondria produce ATP, your cellular energy currency, through a process called oxidative phosphorylation. This process involves a series of protein complexes along the inner mitochondrial membrane, collectively known as the electron transport chain. As mitochondria age and accumulate damage to their own DNA, the efficiency of this chain decreases. Electrons leak prematurely, reacting with oxygen to produce superoxide radicals, one of the most destructive categories of reactive oxygen species. This oxidative stress damages mitochondrial membranes, further impairing ATP production, triggering what researchers call the vicious cycle of mitochondrial aging. The cells compensate by signaling for mitochondrial biogenesis, the creation of new mitochondria, through a master regulator called PGC1 alpha. But in sedentary individuals with chronic inflammation and elevated insulin, PGC1 alpha activation is blunted. The practical consequence is what many of my patients describe as a kind of fatigue that sleep does not fix. The cells are not producing enough ATP, the brain is not receiving adequate metabolic support, and the body has entered a state of chronic energy conservation. 3 interventions reliably upregulate PGC1 alpha and restore mitochondrial function. First, resistance exercise, particularly eccentric loading, triggers a downstream signaling cascade through AMPK and SIRT1 that stimulates new mitochondria. Second, time-restricted eating suppresses mTOR, disinhibits autophagy, and triggers a selective clearance of dysfunctional mitochondria called mitophagy, essentially removing the damaged units and making room for healthier ones. Third, targeted supplementation with creatine monohydrate at 3 to 5 grams daily provides an additional phosphate buffer for ATP regeneration and has been shown in multiple randomized trials to improve functional strength and cognitive performance in adults over 60. Second, time-restricted eating suppresses mTOR, disinhibits autophagy, and triggers a selective clearance of dysfunctional mitochondria called mitophagy, essentially removing the damaged units and making room for healthier ones. Third, targeted supplementation with creatine monohydrate at 3 to 5 grams daily provides an additional phosphate buffer for ATP regeneration and has been shown in multiple randomized trials to improve functional strength and cognitive performance in adults over 60. Stay with me, because I am about to tell you about autophagy, the biological recycling system that Nobel Prize-winning research has called the most important anti-aging mechanism yet discovered. In 2016, the Nobel Prize in Physiology or Medicine was awarded for the discovery and characterization of autophagy. The Nobel Committee described it as a fundamental process for degrading and recycling cellular components. I want to explain why this matters far more than most people realize. Autophagy, from the Greek for self-eating, is the process by which cells identify damaged or dysfunctional components, misfolded proteins, oxidized organelles, fragments of dead pathogens, and systematically dismantle and recycle them. Think of it as a cellular quality control team that continuously removes the biological debris that, if left to accumulate, contributes to inflammation, cellular senescence, and neurodegenerative disease. The key regulator of autophagy is mTOR, mechanistic target of rapamycin, a nutrient-sensing kinase that acts as a gatekeeper. When mTOR is active, which occurs in the presence of amino acids and insulin, autophagy is suppressed. When mTOR is inhibited, during fasting, caloric restriction, or intense exercise, autophagy is upregulated. This is why the pattern of eating matters as much as the content of eating. Continuous grazing, even on nutritious foods, keeps mTOR perpetually activated and autophagy perpetually suppressed. Compressing the eating window to 8 to 10 hours with a clean fasting period overnight allows for significant autophagy induction without the physiological stress of prolonged fasting. Research published in the journal Cell Metabolism demonstrated that time-restricted eating, even without caloric restriction, significantly improved mitochondrial function, reduced inflammatory markers, and extended healthy lifespan in animal models. Human trials have replicated the metabolic benefits, including improvements in insulin sensitivity, blood pressure, and body composition. Polyphenol compounds, particularly spermidine found in wheat germ and aged cheese, and urolithin A, a metabolite produced by gut bacteria from elagitannins in pomegranates and walnuts, have both been shown to directly induce mitophagy through pathways independent of mTOR suppression, offering an additional avenue for individuals who cannot extend their fasting window. If the science I have been sharing is giving you a new way to think about your own health, a sense that aging is not a fixed destination but a process you have more influence over than you were ever told, then you are exactly the person this channel was built for. Golden Glow Senior exists to bring evidence-based longevity science to the people who need it most, and who are most often overlooked by mainstream health media. If that resonates with you, I would be honored to have you subscribe. Hit the button below and make sure the notification bell is on, because what I am about to share next is not something you will want to miss. There is a category of cell in your body that researchers have begun calling zombie cells, and the name is uncomfortably accurate. These are cells that have reached the end of their replicative lifespan, stopped dividing, and refuse to die. In biology, they are called senescent cells, and they represent one of the most significant and underappreciated drivers of age-related disease. Here is what makes senescent cells so destructive. Rather than quietly entering apoptosis, the clean, programmed cell death that normally clears out old or damaged cells, senescent cells remain metabolically active. They secrete a cocktail of inflammatory cytokines, proteases, and growth factors collectively called the senescence-associated secretory phenotype, or SASP. This chronic secretion poisons neighboring healthy cells, disrupts tissue architecture, and drives the same low-grade inflammatory environment we discussed earlier. Senescent cell burden increases sharply after the age of 50 and accumulates in multiple tissues: visceral fat depots, arterial walls, cartilage, the liver, and the brain. Research using transgenic mouse models has demonstrated that selectively eliminating just 30% of senescent cells significantly extended healthy lifespan, improved physical function, and reduced the incidence of age-related disease. Human clinical trials are now investigating a class of compounds called senolytics, agents that selectively induce apoptosis in senescent cells. The most studied combination involves quercetin, a flavonoid found in capers, onions, and green tea, paired with dasatinib, a pharmaceutical agent used in oncology. However, emerging research suggests that quercetin alone at doses of 500 to 1,000 milligrams daily, particularly in its liposomal form for improved bioavailability, may provide meaningful senolytic activity without the risks associated with pharmaceutical agents. Fisetin, a flavonoid found in strawberries, apples, and persimmons, has also shown remarkable senolytic potency in preclinical research. A 2018 study published in eBiomedicine found that fisetin extended both median and maximum lifespan in aged mice by approximately 10%, results that have generated significant interest in the clinical research community. Hold on, because what I am about to share at the 7-minute mark is the finding that, when I first encountered it, I had to read 3 times before I believed it. It changes the way we think about the relationship between the gut and the aging brain. Here is the finding I promised you. For most of medical history, aging was treated as a problem of individual organs. The heart wears out, the joints degrade, the brain slows. We have spent decades studying each system in relative isolation. but a convergence of research from the fields of microbiome science, neuroimmunology, and epigenetics now points to a single unexpected origin point for much of what we call aging. And it is sitting in your intestines. The gut microbiome, the approximately 38 trillion microorganisms living in your gastrointestinal tract, is not merely a digestive accessory. It is a primary regulator of immune function, systemic inflammation, neurotransmitter synthesis, and epigenetic gene expression. And it ages. In fact, it ages faster than almost any other system in the body. Research published in Nature Aging in 2021 tracked the gut microbiomes of over 9,000 individuals across multiple decades. The findings were extraordinary. People whose microbiome diversity declined sharply in their 50s and 60s showed significantly faster biological aging across every measured biomarker: telomere length, inflammatory markers, cognitive function scores, and cardiovascular risk profiles. Conversely, individuals who maintained high microbiome diversity into their 80s and 90s, the so-called longevity clusters, shared a specific signature: a higher proportion of rare, disease-suppressing bacteria and lower levels of pro-inflammatory opportunistic species. The mechanism involves something called leaky gut, technically increased intestinal permeability. As the microbiome diversity decreases and inflammatory bacteria proliferate, they produce compounds that degrade the tight junction proteins holding the intestinal lining together. This allows bacterial fragments, particularly a molecule called lipopolysaccharide, or LPS, to enter systemic circulation, where they trigger the exact same pattern of low-grade inflammatory activation that drives inflammaging, mitochondrial dysfunction, and cellular senescence. This is the connection most people miss. You cannot fully address systemic aging without addressing the gut environment from which so much of it originates. The most evidence-supported interventions for restoring microbiome diversity include a dramatic increase in dietary fiber diversity, targeting 30 or more distinct plant species per week, the strategic use of fermented foods such as kefir, kimchi, and aged cheeses, and the elimination of emulsifiers like carboxymethylcellulose and polysorbate 80, which a 2015 study in Nature demonstrated directly disrupt the mucus barrier separating bacteria from the intestinal epithelium. We are nearly at the end now, but there is one more mechanism I want to cover before I give you the complete protocol summary. It involves something you do for approximately 1/3 of your life that determines the fate of your cellular repair machinery. Every night, during the deep, slow-wave stages of sleep, your brain undergoes a process that has no daytime equivalent. The glymphatic system, a network of channels surrounding cerebral blood vessels, expands by approximately 60% and begins actively pumping cerebrospinal fluid through brain tissue, flushing out the metabolic waste products that accumulate during waking hours. Chief among these is amyloid beta, the protein fragment associated with neurodegenerative disease. Outside the brain, deep sleep is the period of peak growth hormone secretion, the primary hormonal signal for tissue repair, muscle protein synthesis, and cellular regeneration throughout the body. It is also the window during which cortisol reaches its lowest circadian nadir, allowing anti-inflammatory repair processes to dominate. Disrupted sleep architecture, even without a reduction in total sleep duration, significantly impairs glymphatic clearance, suppresses growth hormone pulses, and elevates inflammatory cytokines the following day. A single night of poor sleep has been shown in research from the University of California to increase amyloid beta accumulation in the brain by a measurable margin. Chronic sleep disruption compounds this effect dramatically. Practical interventions with strong evidence include maintaining a consistent sleep-wake schedule to anchor the circadian rhythm, reducing blue light exposure in the 2 hours before sleep to preserve melatonin secretion, and ensuring adequate magnesium glycinate, 300 to 400 milligrams taken 60 minutes before bed. which activates GABA receptors in the brainstem and has been shown to significantly improve deep sleep duration and sleep efficiency in older adults. Before I give you the complete protocol summary, I want to say something directly. Everything I have shared with you today, the Inflammaging research, the mitochondrial science, the gut-brain axis findings, none of this was part of my medical school education. I had to seek it out, study it independently, and then watch it transform the way I think about health for the people I care about most. That is why Golden Glow Senior exists. If you are serious about living longer, feeling stronger, and thinking more clearly in the decades ahead, this channel will keep giving you the science you need to make that happen. Subscribe now and bring someone you love with you, because this information does not belong locked behind journal paywalls. It belongs with you. Let me bring this together. We have covered 6 interconnected mechanisms that drive biological aging: chronic low-grade inflammation through the NF-κB pathway, mitochondrial decline and the suppression of PGC1 alpha, the accumulation of senescent cells and their toxic secretory phenotype, the Nobel Prize-recognized process of autophagy and how its suppression accelerates cellular aging, the gut-brain aging axis and the profound role of microbiome diversity in systemic inflammatory burden, and finally, the glymphatic system and the irreplaceable role of deep sleep in cellular repair. None of these mechanisms operate in isolation. They form a network, and intervening on one node tends to produce measurable improvements across the others. That is the reason the evidence-based protocol I follow personally targets all 6 simultaneously rather than addressing each in sequence. The core takeaway I want you to carry from this video is this: your biological age is not fixed. It is not determined entirely by your parents' genetics or by the decades you have already lived. It is a dynamic, ongoing process, and the scientific literature is increasingly clear that the interventions required to influence it are not extraordinary. They are accessible, they are actionable, and for most adults over 50, it is genuinely not too late to begin. I am Dr. Linda Harrington. Thank you for watching. If this video helped you see your own biology differently, share it with someone who needs to hear it. I will see you in the next one.